Hola amigos, bienvenidos a este nuevo espacio de investigación y capacitación acerca de los ensayos clínicos más avanzados en el mundo de la ELA. En este caso, la investigación de Tudca, un compuesto que tiene su base de investigación en Europa, más precisamente en Italia, y invitamos para que nos pueda ilustrar al respecto a la doctora Antonia Angela Cocco, a quien presentamos a continuación.
Good morning, everyone. I'm Antonia Angela Cocco. I'm a neurologist working at the Humanitas University Hospital in Milan, Italy, in the team of Professor Albanese, who is the professor who led this study. So I'm going to present you this trial, a phase 3 trial, which evaluates the safety and efficacy of tauroursodesoxycholic acid, Tudca, as add-on treatment in patients affected by ALS. As you can see, this project was funded by European Commission within the Horizon 2020, which is a research and innovation program of rare disease and orphan drugs. So this is a phase 3 multi-center randomized double-blind placebo-controlled trial. So understanding the molecular and biochemical pathogenesis of neurodegenerative disease is very important in order to discover neuroprotective therapies that aim to have a disease-modifying effect. As you can see in this you know that ALS is one of the most fast progressive neurodegenerative disease, and it affects mainly the corticospinal and spinal muscular neurons. As you can see in this slide, there are several pathways that, which are involved in motor neuron death. For example, so there is a role of excitotoxicity, oxidative stress, but also mitochondrial dysfunction, and also the accumulation of misfolded proteins, the apoptotic pathways, and there is also a role of neuroinflammatory, of neuroinflammation. So there have been repeated attempts to develop medication which have a potential disease-modifying effect. Currently, there are only 2 compounds with such activity that have received marketing authorization. One is Riluzole worldwide, and the other is Edaravone in a limited number of countries. So modifying disease progression is a realistic goal. Why Tudca? Tudca is tauroursodesoxycholic acid, and with Udca, which is ursodesoxycholic acid, they are hydrophilic bile acid, which are normally produced endogenously in humans in the liver. In particular, Tudca is the taurine conjugated form of Tudca, which is more hydrophilic, and this is important because it is able to cross the blood-brain barrier. We know this compound since a long time because it has been administered for months to years to patients with a variety of liver disease. And what we know about this compound? We know that it has a very good safety profile because the mostly reported adverse events are gastrointestinal, such as nausea, diarrhea, abdominal pain, and in few cases also rashes and pruritus. Another important thing is that at present, no contraindication to the use of Tudca are known. So, Most of the pathways on which Tudca exerts its anti-apoptotic and protective, cytoprotective properties have been studied in hepatocytes. But we know that similar mechanisms are also present in other types of cells, including neurons. So, Tudca exerts its effect by acting as a chaperone, and it reduces protein aggregation and deposition. This led also to a reduction of reactive oxygen species, and so ultimately leading to protection against mitochondrial dysfunction. It is also able to ameliorate endoplasmic reticulum stress. And we know also that it inhibits the expression of pro-inflammatory cytokines, and so this it has also an anti-neuroinflammatory effect. Finally, it is able to regulate the expression of some genes which are involved in the apoptotic cascade. In particular, regarding these, caspases are a family of protease enzymes playing an essential role in programmed cell death. In particular, high activity of caspase-3 has been detected in patients with ALS. And there are studies in vivo and in vitro that support the view that both UDCA and TUDCA have cytoprotective properties by inhibiting caspase-3 activation. Regarding the clinical evidence of the efficacy of TUDCA, we start before with another trial, this pilot trial. It was a phase 2, in which 3 different doses of UDCA were administered to 18 patients for a period of 4 weeks. This study was very important because it provides relevant findings on the pharmacokinetic profile. In fact, in this study was shown that UDCA was able to cross the blood-brain barriers, and it was detected in the cerebrospinal fluid in a dose-dependent manner. However, this study had some limitation. For example, the short treatment duration, and so it was not possible to make a conclusion about its efficacy. Regarding TUDCA, the efficacy of this compound was studied in this phase 2 trial, in which 34 patients were randomized to placebo or TUDCA, 1 gram twice a day, sorry, for 54 weeks. What was this study design was innovative because the patients were assessed during a lead-in period. This provided the opportunity to measure the natural disease rate progression and to compare it to the period in which the patients were taking the experimental treatment. So in this study was performed a categorical analysis. In fact, were considered responders patients who had at least an improvement of 15% in the ALS functional rating scales revised slope during the treatment period compared to the lead-in period. So it showed that the number of responders was much higher in the Tudca arm compared to the placebo one. So a second phase 2 trial evaluated the efficacy of a co-formulation of Tudca and sodium phenylbutyrate. This is another compound which is able to reduce the endoplasmatic reticulum stress. In this trial, the patients were randomized to this compound once a day for 3 weeks and then twice a day for a total of 24 weeks with a randomization 2-to-1. Also, in this study, it was shown that there was a slower functional decline using ALS functional rating scales in the active drug combination group compared to the placebo one. However, there were no differences in other secondary endpoints such as muscle strength or respiratory function, and also they did not find any differences in neurofilament levels. So in our trial, this is a phase 3 trial, so it was planned to enroll 440 patients, and the primary objective of the study was to assess the efficacy of Tudca in slow disease progression as measured by ALS functional rating scale revised. The secondary objective of this study were evaluated the survival time. There were also some questionnaire about quality of life, forced vital capacity, and muscle strength. Then another one was to assess the long-term safety and tolerability of Tudca at this dosage for up to 18 months in these patients. So as you can see, this trial was conducted in several centers in Italy and in other European countries. As I was saying, it was planned to enroll 440 patients. However, due to the pandemic, at the end, we were able to enroll only 330 patients. As you can see, the inclusion criteria in this slide. So the patients should have a diagnosis of ALS according to LS-Corial diagnostic criteria, a disease duration less than 18 months. They should not have any swallowing difficulties, and they should been able to perform a spirometry. Another inclusion criteria, in fact, was forced vital capacity more or equal to 70% of the predicted one. And also, they should have been on Riluzole treatment. They should have been stable on Riluzole treatment for 3 months in the lead-in period. Patients who were taking Darabon were excluded because, as you know, it is not yet approved by EMA. So, as you can see in this slide, the study design was very similar to the phase 2. There was, in fact, a lead-in period of 3 months, and then the patients were randomized with a ratio 1-to-1 to 2 grams of Tudca or to placebo. Why so as I was explaining, there was also in this study a lead-in period. Why this lead-in period is so important? Because the assessments made during this running period provide the best prediction of disease course for each patient, and this helps to reduce interpatient variability. This lead-in period is suited to linear efficacy measures, such as the ALS functional rating scales, for which the entry level and slope are predictive of survival. Also, in this study, we are going to perform a categorical analysis. Based on the suggestion received by EMA, the threshold to consider a patient responder was increased to 20%. So are considered responders patients who have more or equal 20% change in ALS functional rating scales slope between lead-in and treatment periods. Also, we are going to perform a post-hoc analysis using a threshold of 25%. As you can see in this slide, so the patient were performed several clinical and laboratory assessments during all the study. But another important aspect of this study is that we are going to evaluate the efficacy of Tudca on possible biomarkers. As you maybe know, neurofilament levels are considered a potential biomarker of neurodegenerative disease. We know that their levels are higher in patients with ALS compared to controls, but compared also to other neurodegenerative diseases, such as Alzheimer's, for example. However, even though they are considered now a measure to evaluate the disease progression and survival, there are no data about if they are useful, for example, to monitor the therapeutic effect. So one of our approaches is to combine several biomarkers. For example, in this study, we are not only going to measure neurofilament level in cerebrospinal fluid and serum, but also plasma creatinine, because it was recently reported as a robust biomarker of disease progression. It appears to be a strong and independent predictor of mortality in ALS, and it is not influenced by site of disease onset. Moreover, it is comparable to the ALS functional rating scale score. And it is the measure of plasma creatinine is simple, is cheap, and widely available. Another biomarkers that we will evaluate is Metalloproteinases 9. This has been found elevated in tissues and biofluid of ALS patients. And we know also that elevated concentration of these metalloproteinases in CSF and serum were associated with muscle denervation and extensive neuroaxonal degeneration, causing motor neuron loss. We know also that Tudca it is able to inhibit these pathways. So this is very useful also to evaluate the therapeutic effect. If you have any question, I have finished. Thank you for your attention.
Well, thank you so much, Dr. Coco. That was excellent. Thank you so very much. That was very comprehensive, and I think it was easy to understand, which is important for all those patients and families that are watching this presentation and this interview. So I just had a couple of questions for you that I think pretty much express what some people express the doubts that some people may have about the Tudca trial.
Yeah.
And one of those questions is related to another trial that is taking place in actually many parts of the world, but especially in North America, which is the trial of Amylyx, AMX0035.
Yeah.
So one of the two compounds that are being evaluated in the AMX trial is precisely Tudca. So I guess a very natural question would be, how would Tudca, the compound that you are studying, in case it gets approved, it becomes approved, would combine with AMX0035, also in case AMX0035 gets approved as well?
So, I mean, there is also the possibility to use both. I mean, because this disease is very complex, so maybe in the future we will use more drugs in order to inhibit different pathways and to control better all these mechanisms. So I think it's always a good news if a drug shows an effect on ALS. Maybe like in other diseases in the future, we'll take more, we'll suggest the patient to take more than one drug.
Right. So what we are hoping for is to have a cocktail of different drugs, which could be Riluzole, Edaravone, plus AMX, plus Tudca. The best scenario would be that a patient can take the 4 compounds or even more compounds as some kind of cocktail.
Even though I'm convinced that it's important to show that they have a real effect. This is the reasons why we are using this categorical analysis, because we want to evaluate if the improvement is significant. Sorry.
Great. Excellent. So the other question would be also related to AMX. Is there any difference between the taurusordiol that you are using, the supplement that you are using, and the one that they are using in the Amylyx trial? Or is the same compound?
Now, about the other compound, we have tried to compare the slope of the 2 studies, the phase 2 trials, and the one only with Tudca. And we saw that they are very similar, the slope. So now it's important to understand if there is an advantage to add also sodium phenylbutyrate. because there were only trial testing the safety of this drug, but not really its efficacy.
And considering the information you have so far, would you say that the combination of those two compounds works better, or it looks like Tudca works better by itself? Or maybe you cannot make any conclusion about that yet?
I think that we have to wait for a bigger trial for the other compound. I mean, with a higher number of patients. Great,
I understand. So then the other aspect of Tudca is that it is a supplement. It is a supplement that you can buy in any pharmacy in general, in many countries. So again, what would be the difference between the Tudca compound that you can find in any pharmacy over the counter and the one that you are using for your trial, if any, if there is any difference? And on top of that, what would you recommend to people who buy this product on their own in pharmacies?
Well, I think that it's important to wait for the results before to suggest to the patient to take any type of drug. Then also there is no any important difference between the compound that we use in our study and the one that is usually taken for this liver disease and so on. This is the same company in Italy that furnished this drug to us. I don't know then in other country.
Okay, I understand. It's pretty much the same compound, but you say that people should wait before taking any kind of supplement or medicine on their own without having the scientific monitoring.
Yes.
Mm-hmm. That would be your recommendation.
For sure.
So when are we?
Even though, as I was saying, it's a very safe drug.
It's a very safe drug.
Yeah.
Okay. Okay. When are you expecting next results in this Tudca trial?
So at the end of these years, maybe we will be able to give you the results, because now we are performing all the analysis, all the statistical analysis, and also there are some patients who are doing the open phase of this trial. So we will have all the data at the beginning of the next year, maybe.
And once you have the results of this phase 3 trial in December or January, what do you expect to be the next stage besides the open label extension stage? Would you expect approval from EMA, or would you expect another trial with more participants?
No, I think the number of patients, even though it was less than the expected one, but I think it will be sufficient to make a decision about the efficacy of this drug.
So you would say the number of participants that have been recruited for this phase 3 should be sufficient for approval in case the results are successful, of course.
Yeah, I hope. Yes, but our statistic told us that it will be enough, so we will see.
And how many participants are involved right now in this trial?
So we have enrolled 330 patients. So it's a high number, I think.
In how many different sites in Europe?
So as I was showing you, I don't remember in total, but only in Italy we have 7 centers, and then we have some centers in UK, Ireland, and Germany and Netherlands. So they come from different European countries.
So you're not expecting, as of now, at this point, you're not expecting to expand this trial to other parts of the world, to other sites?
No, for now, no.
Not for now, okay. Well, hopefully we are really crossing our fingers that this trial is successful, and we are also crossing our fingers for the AMX trial, of course, and any other trial. So I think, yeah, I think that's pretty much everything we can ask for. I think those are the questions. And do you want to also do you want to tell us a brief description of this Tudca consortium that has been formed in Europe? How the consortium works, how it started, in brief, of course.
So it started, I mean, in 2018-19, and they have been involved most of the important centers which follow patients with ALS. And I think that all the centers, even though there were all the difficulties of the pandemic, were they tried to recruit as many patients as they could. So I think we had good results from these collaborations.
And Dr. Albanese is the one who coordinates.
Yes, he's the one who coordinates the studies and the one who proposed to study this drug for ALS.
Mm-hmm. So lastly, tell us a little bit about yourself. When did you get involved with ALS, MND, and how did you get involved with this Tudca trial?
So I was involved 2 years ago. I'm usually working in the field of movement disorders, but then Professor Albanese proposed this trial, and so I was the neurologist to follow the patients in my center. And I think it's a very fascinating disease, and I mean, also it was very important for me, the relationship that you create with the patients, because this disease, unfortunately, is one of the most fast progressive, also compared to other neurological diseases that I see usually in my field. So I mean, it was a great experience for me.
Great, and we appreciate that you are getting involved in this disease, and that you work every day to find a better treatment for this disease that is so severe. Okay, any message that you want to share with the community in Argentina? You know, we have a very vast Italian community in Argentina, like almost around 50% of our population is Italian descent.
Yes,
including myself. But all our videos and all our materials are also seen by all the Spanish speakers around the world, not only in Argentina. So any message that you want to share with all of them, with our patients and families?
Yes, I think that is, I want to share an hope message, because I think that there are a lot of studies now. So a lot of people, a lot of researchers are focused on this disease, and we really hope in the next years to find a cure, some drugs which are really efficacy for this drug. I think that this is a good moment for the research in this field, and I hope that we will see the results very soon.
Okay, that's great. Thank you so much, Dr. Coco, for everything.
I want to greet all the patients and their relatives, and it was a pleasure for me.
Thank you so much, and thanks for doing this in English. I'm sorry that we don't speak Italian here. We know some words, but we don't speak Italian. But you did really well. You did really well. Okay. And thank you again. I want to say thank you to you and to Dr. Albanese as well. So please send our greetings to him. And okay, and just keep working for ALS. Keep working for ALS, and there people has a lot of hope nowadays. I mean, there are so many clinical trials going on around the world, and
absolutely.
Hopefully Tudca will get approved by the end of this year, or maybe the beginning of by the end of this year, or maybe the beginning of 2023. So thanks again, Dr. Coco. Thank you so much for your contribution.
Thank you. Bye, everyone.
Okay, bye-bye. Thank you so much.